Hi, everyone. Welcome back to class. Today is the 7th of October, and we are going to jump right into things. I will be up front. I apologize that I forgot to grade your homework, and that is not fair. I will do that. And like I said, um, I will not be assigning a new homework until that's done. And I will hopefully have that assigned homework very quickly. Another thing that I wanted to mention was that I've added some links to our Canvas page so that it's easier to toggle back and forth between old stuff and new stuff. Um, Chat. Hello. I had a question about the next exam around how many questions would be on exam two about exam one topics. Okay, great. It's not too early to ask. Don't worry about this. Like the question that was posed, this private message was number of questions on exam two that reflect exam one material. And I would say that if the exam has 40 questions total, then you're probably looking at about 10, maybe 11 questions that relate to exam one, and the rest of it will all be exam two stuff. Okay, I hope that I hope that clears that up. I am going to go back to where we were. So if you remember last week in class, what we finished on Thursday was we were talking about enzymes. But if you also remember, I was kind of like, hey, enzymes, here we go, here we go. I'm going to take a step back because there were the last few things that I wanted to talk about with respect to hemoglobin and myoglobin. We talked about this compound right here, bisphosphoglycerate, BPG as it's commonly known. Now, BPG is an allosteric effector of hemoglobin in that what it does is it's going to influence the T or R state of hemoglobin as being the form that we find. Now, this slide, I went over this one a little bit, and so I'll do it again today. This is really just to show how different things contribute to the P50 of hemoglobin. And one thing that I want you to notice is that when hemoglobin is in the absence of bisphosphoglycerate, carbon dioxide, and anything else that's found in the whole blood mixture, well, hemoglobin has a P50 of approximately 10 or so. I don't need you to remember that name, or that number, but what I do want you to take away from this is that CO2, BPG, both will independently cause the P50 to increase. Therefore, the affinity that hemoglobin has for oxygen will decrease in their presence. They have an additive effect because, as you can see here, this is just CO2, this is just BPG, but in combination, well, they both shift to the right. Now, whole blood has a P50 that is higher than that, indicating that there's something else in the mixture, and not necessarily one singular thing, but your whole blood P50 is impacted by both BPG and CO2 as well as other factors. Now, with respect to bisphosphoglycerate, one of the things that I wanted to mention was is bisphosphoglycerate favoring, I'll say favors the, is it, could anyone tell me in the chat, is it the T or the R state of hemoglobin? Is it the relaxed state, the functional state, or is it the T state, the tense state? Well, bisphosphoglycerate, you're absolutely right. Bisphosphoglycerate is going to favor the T state. Because remember, bisphosphoglycerate kind of inserts itself in the center of the cavity, that central cavity, which I will show right here. And it kind of like jumps in there and locks it all up. So it stabilizes the T state. So it might be like, oh, well, bisphosphoglycerate, that's not a good thing. You know, if you, I have a tendency to think about things as either like positive or negative. Is this a good thing or is this a bad thing? And I would think, well, favoring the T state, that's got to be bad. That's not good. I mean, favoring the form of hemoglobin that does not bind oxygen, I don't want that. Well, there is an instance where it actually is kind of a good thing. So this graph right here is an oxygen binding curve. And what I want you to notice is that we have a P50 right here of our venous hemoglobin. That's approximately, I'm going to call that 23, we'll say. Or yeah, 23, 22. That's our P50. Now, in the presence of BPG, we see it shift up to almost 30. Okay, so our P50 goes up. Now, what's going on in this slide is kind of interesting, and in this graph is pretty interesting. So atmospheric pressure decreases with altitude. So as if you go to Denver or something like that, um, Denver is the mile high city. So if you're at 5,280 feet or higher than that, there's a little bit less oxygen in the air than if you're at sea level in San Marcos. Now, the short-term impact of that, well, your body's going to respond by increasing the production of BPG. Give me a one in the chat if you think that's a good thing, or two if you think it's, wait a minute, that doesn't make sense. And so the increase of BPG. Now, my first instinct whenever I saw this, I was like, that can't be good. Because if there's not much oxygen, well, then I want to avoid BPG because I want my hemoglobin to be able to bind as much oxygen as there is. And if there's less oxygen, that compromises it. Now, with that in mind, this is the sort of slide that any instructor will show and be like, you wouldn't think this. And then it's like, you're right, I wouldn't. And then at the end of the slide, it's like, but actually, it's this. And it's like, oh wow, it's amazing storytelling there, Dr. Gray. You're really great at this. Well, this is kind of capturing that where bisphosphoglycerate does have a positive impact. And here's what's kind of tricky. What I want you to notice about this black line is this shows essentially under normal conditions, about 38% of the oxygen that is picked up by hemoglobin will be delivered to the tissues. So if you think about that in terms of, I always kind of think about it in terms of like individual hemoglobin molecules. They're dropping off between two oxygens and one oxygen. About 38% of the oxygen that's taken in by your body gets deposited to the tissues. Okay. So what that means is that when hemoglobin is leaving the tissues, about 62% of O2 remains. Give me a Y or an N in the chat. A Y if that makes sense, and N if that does not make sense. Okay. So under low atmospheric conditions, well, we deliver about 30%. Oh, wait a minute. Okay, so under low atmospheric conditions, we're delivering even less. So that same like floor. So in the absence of BPG, you're still only, or you're still, all of the hemoglobin that's leaving your tissue still has about 62% of the O2 intact. But the difference is that it didn't get to 100%. Could everyone give me a Y in the chat if that makes sense? Okay, so hemoglobin, when you're in Denver, hemoglobin in your body can't get to 100% saturation or very close to that. Instead, it gets to approximately 92% saturated. Can you give me a 5 in the chat if that makes sense? Okay. Now, what causes a little bit of a shift is that bisphosphoglycerate gets produced by your body in response to this, well, this lower amount of oxygen. And what will happen is hemoglobin in the presence of bisphosphoglycerate will bind less O2, but it will ultimately drop off more O2. So there's less oxygen there, and hemoglobin binds less oxygen, but it drops off more. Okay, so BPG in some circumstances is arguably a good thing because it allows hemoglobin proteins to drop off more oxygen at high altitudes. Okay. Can everyone give me a 9 in the chat if that makes sense? A 1 in the chat if they want me to go over it again. Okay, so the point of this is BPG at, I'm going to say point up altitude helps HB or hemoglobin drop off more O2 at tissues. That's the takeaway from this. That's what BPG does. Sure, it binds less O2, but it drops off more. Okay, at higher altitudes. Yeah, my arrow pointing upward was elevated. Let me change that. BPG at elevated altitude. Okay. Can everyone give me a 7 in the chat if that makes sense or makes more sense? Okay, cool. So with that said, um, okay, well, I think that it might be. Okay, so I got a private message question that was, is this because it happens faster? And I interpret that to mean, is this because you get to this saturation or the lower saturation point more quickly? I think ultimately it has to do with the fact that more of your protein is in that T state than it is in that R state. And so basically you have less oxygen to possibly bind, and so it can release, it has like a lower threshold. I hope that makes sense. So in essence, yeah, it might happen faster. But I'm just kind of cautious about saying faster or talking about rates because that gets us into a discussion of like how quickly does one particular step take place. Um, okay. So this slide right here is very important. Shifts to the right. What are things that cause our hemoglobin to shift to the right? What causes it to shift to the left? Increase in pH will cause a shift to the left. Well, how is the pH going to increase? Well, it's by hydroxide ion concentrations increasing because hydrogen ion concentrations are decreasing. Well, an increase in hydrogen ion concentrations will cause the pH to go down, which will shift toward the T state. So I interpret this to mean a shift to the right is the same as a shift to the T state. A shift to the left is the same as a shift to the R state. Now, one other hemoglobin that I wanted to mention was fetal hemoglobin. Now, fetal hemoglobin, also known as HbF, well, fetal blood has a higher affinity for the mother's or O2 than the mother's hemoglobin does. Now, why fetal hemoglobin is different in structure. Rather than being a pair of alpha and a pair of beta subunits making up the tetramer, instead, fetal hemoglobin has a slightly different composition. It's got two alpha subunits and two gamma subunits. So alpha 1, alpha 2, gamma 1, gamma 2. All that I want you to take away from that is that these two subunits that make up that are different from the beta subunits. What about them makes them different? In terms of the sequence, it's probably not much. It's probably like 90% almost identical. The intrinsic oxygen affinity for HbF is very similar to adult hemoglobin. So HbA, adult or the maternal hemoglobin. Now, the HbF has a lower affinity for BPG than does HbF. What do you think that means? Well, if you have a lower affinity for BPG, that means that most of HbF is in the R state. And let me add in there more HbF is in the R state at the same oxygen concentration as if the HbA were present. So think about it as if you had HbA and HbF and you put them in one environment, a higher percentage of those proteins that are HbF are going to be in the R state, the active or the oxygenated form. What accounts for this difference? Well, as it turns out, there is a mutation, and not necessarily a mutation, but there's a amino acid difference between them. Adult hemoglobin A has a histidine at position 143. I don't want you to remember that specific residue, but this leads us into our discussion on the next slide. HbF has a serine at that position. Now, this is one of the things that's important about amino acids. How would you classify these two amino acids? Are they N nonpolar or P for polar? They're both P, polar. Okay. Histidine. What do we know about histidine? Would we, as a polar, would we classify it as U for uncharged, P for positive, or N for negative? Histidine is a positively charged amino acid. So it's going to be a polar positive residue. What about serine? How do we classify that? As a polar, P, U uncharged, or N negatively charged? It's an uncharged amino acid. So they're both polar, so there's going to be some similarities in how they behave. But one fundamental difference is that histidine can participate in ionic interactions. Can serine participate in ionic interactions? Nope. Can it participate in hydrogen bonding? Yeah, absolutely. Histidine could potentially, but if histidine is protonated, the interactions are going to be different. So the point that I want you to take away from this is that the types of interactions that different amino acids have are very important to think about. Now, the charge or the change of a positively charged histidine for a neutral serine reduces the binding affinity of BPG to HbF. So there we go. Okay, now the last slide in this slide deck is all about abnormal hemoglobins. These are what you would kind of appropriately, not appropriately, but you could refer to these as abnormal or mutant hemoglobins. Now, one of the things that I want you to notice about the names of these is that lots of them are locations. For instance, Philly, Boston, Milwaukee, Yakima is in Washington, I know that. And Hammersmith, I think they mentioned that in the cartoon Bluey. Can't remember. My kids have watched it. Anyway. So I think that might be in Australia. I could be wrong. At any rate, these are mutations that are kind of naturally occurring in hemoglobin that you've maybe never heard of anyone having. That doesn't mean that they aren't, a part of the population, but they don't have as dramatic of an effect as something like sickle cell anemia. I want you to think about sickle cell anemia as being one that's very similar to these. Now, when it comes to this mutation, this is sort of a notation that I want you to know about. What this is showing us is that we have a phenylalanine, that's the wild type mutation, in the CD1 position. So that's when you think about the structure of this, amino acid number 42 in the beta subunit has been mutated to a serine. I'm not going to ask you to decipher all of these, but one of the things that I do want you to know is that a little bit more commonly, you'll see a mutation like this indicated in the following way. F for the single letter code of the wild type amino acid, then 42 being the location of that amino acid residue, and then S for the new residue that is in that location. So what I want you to look at, and the way that I would ask a question about this sort of thing would be, hey, here's a mutation. Tell me about the classification. How does the classification of these amino acids change? In this case, we have a tyrosine, a polar uncharged residue that's mutated to a phenylalanine. So we've got polar uncharged to a nonpolar residue. I want you to decipher that. That's one of the ways that I will take exam 2 material and ask an exam 1 type of question about it. I hope that makes sense. Could everyone give me a 5 in the chat if they understand that's the sort of question that I would ask about this? Okay. Now, with that in mind, these mutations, well, they can have a wide variety of impacts. Some of them are going to be, well, here we go, an aspartate mutated to a histidine. We disrupt a hydrogen bond that stabilizes the T conformation. Well, with that sort of consequence, that person's If you have the Yakima mutation, chances are most of your hemoglobin is going to be in the R state. Or maybe there's a little bit more that's in the R state. So maybe that means that your P50, well, your P50 might be lower. Um, So, mutations may alter hemoglobin's quaternary or tertiary structure, destabilize the hemoglobin to the extent that the red blood cells cause lysing or hemolytic anemia, and they might favor one of the different states of iron. It might favor the 3 plus or the iron 3 structure or iron 2 as opposed Iron 3 as opposed to Iron 2. Again, lots of this information is just kind of on this slide is a little bit more trivial than anything else. Um, and this is something that if you are planning on going to medical school or something like that, this would be a good slide to be like, oh, okay, I've got my connection between the molecular and the physiological. Okay, there's my connection where I have mutations that, well, it's a single amino acid that's been switched from a tyrosine to a phenylalanine. Okay, what does that mean? Disrupts hydrogen bonding. Okay, so that's at the molecular level, and I don't know the effect in terms of the physiology, but this might be something where, somebody struggles with like they have to live at lower at sea level, as opposed to, could be like extreme illness at altitudes. Okay, now where do we go from here? So where we go from here is I'm going to stop my share. I'm going to end that slideshow and I'm going to go on to the next one. Discard. Now, where we are is we're going to get into enzymes. Now, enzymes are one of my favorite things to talk about in this class. Um, and the reason is because to me it makes a little bit more sense. So what I'm on is the slide deck that's titled Week 6, Day 2. So hemoglobin is to me something that's like, oh, I guess I kind of understand it. I can follow it. It's a, protein. It has a function that it grabs something and then it releases it. Okay, cool. I always think about enzymes as being like a protein that grabs something and then makes something new. And to me, it's a little bit more arguably kind of interesting. Now, with that in mind, we I always talk about protein function and ligand or protein ligand interactions before I talk about enzymes because there are some things that are true of proteins like hemoglobin that are true of enzymes. For instance, there are enzymes that have the T and R states. Proteins that catalyze a reaction that goes from or catalyzes a protein will, or an enzyme will change from the T state to the R state. Now, these first few slides were all about, hey, how quickly does a reaction go in the absence of an enzyme? You might have something that takes 78 million years to be completed in the absence of an enzyme. Well, in the presence of an enzyme, it can be done in a matter of seconds. And so we're seeing this sped up 1 times or 1.4 times 10 to the 17th. These specific numbers, are they ones that I want you to know? No. Do I want you to have a sense of how quickly a reaction can be expedited? I do. I do want you to understand that all of these reactions, well, and like I gave you the answer options of, like on the first poll question, which I'm going to go ahead and give this one a, nope, nope, nope. Where did it go? Where did it go, Joel? We're not seeing a reaction speed up 10x. We're not seeing it speed up 1000x. 7.7 times 10 to the sixth, we're seeing it speed up almost a million x. So an enzyme, if there was an enzyme that sped up a reaction 10x, it's not really holding its up end of the bargain. It's not really doing that much more impressive. Oh, I'm sorry, I missed a note. Should we know the effects to predict the RT states of or P50. Um, I'm going to go back to that slide just on here on my own computer. Um, if that was in reference to the abnormal hemoglobins, I don't want you to know. No, don't. I would not stress about those details. Instead, I want you to know the general impacts of what causes a shift to the R or the T state. So, what are the kind of environmental conditions rather than the mutations? The mutations, like I said, those are things that um, if you're going to go to medical school, it'll be good to know that to discern between something like sickle cell anemia. Like there are some mutations in hemoglobin that are not kind of potentially fatal and that do not require dramatic or significant intervention. Okay, so we've got our enzymatic reactions. If there's an enzyme that speeds up a reaction 10x, it's not that impressive. No offense, enzyme, but there we go. Now, what is an enzyme? An enzyme is a biological catalyst. Does anyone want to, or could somebody add to the chat a catalyst that you've talked about that you talked about in organic chemistry? I always think of the Grignard reagent. Not necessarily substrate. No. Whenever I think of I'm thinking like the Grignard reagent or NABH4. These are compounds that whenever you add them to the reaction, they speed them up. Now, a substrate, that's a term that we're going to use in just a little bit when we talk about enzymes. H2SO4, sulfuric acid, yeah, that can act as a catalyst. Um, those would be perfect. These are all great. Thank you very much. So a biological catalyst, well, when we're talking about an enzyme is a biological catalyst because it's going to take a reactant and convert it to a product or assist with the reactant becoming the product. And what makes up the molecule that does that is its biological in origin. So a biological molecule that, simply put, helps a chemical reaction take place. And in the process, speeds it up quite a bit. Now, which of the following is a key element of a chemical reaction? I want to see poll question going up now. We've got movement of electrons or making and breaking bonds, destroying atoms, making atoms, or creating water from O2. Okay. So what I was really looking for here, which we're at 94%, I put this out here to kind of shift you in a specific direction. Um, can a chemical reaction make an atom? No. Can it destroy an atom? No. And then creating water from O2, well, that's also kind of making an atom because water requires H2O. And so we can't just kind of spontaneously let that happen. So really the best answer and the one that I was looking for was answer option A. And we ended up with about 93% of the class selecting that. The other answer options were split between B and C. Now, A is the answer that I was looking for there. All right. So if an enzyme is going to help speed up a reaction, what is that thing, that biological molecule or that molecule that's biological in origin? Yeah, an enzyme is a molecule. Water's a molecule. So is water an enzyme? No. Is an amino acid a molecule? Yeah. Is a dipeptide a molecule? Answer that. So would everyone agree? Add a 1 to the chat if you agree that an amino acid is a molecule. And a 5 if you dissent. Okay, so if we've got something like aspartic acid as a molecule, then let's say that we have aspartic acid D. and glutamic acid, a dipeptide, DE. Add a 5 to the chat if that is a molecule, or any other number if you don't believe that it is a molecule. Okay, cool. Um, then is a polymer of 453 amino acids a molecule? Add a Y or an N to the chat. Yeah. So we started out this discussion, an amino acid, that's a smaller molecule, and then we made it a larger molecule as an example. And yeah, that's also true. It is a molecule. And then we had oh boy, if a two amino acid molecule is a dipeptide, a 453 amino acid molecule, I don't know what that is, but that would be a 403 AA peptide. So yeah, this is a molecule, and the same thing goes here. Yes, these are all molecules, and these are molecules that are produced within cells. Now, if you understand this, that's a very important thing for you to understand. Amino acids, why wouldn't it be a compound? That's a great question. I'm not really going to discern between the two of them. I think in this case, um, they would be kind of interchangeable. It's a fair question, though. Um, so you could refer to it as a compound because it does kind of fit that definition. I'm just going to default to refer to them as molecules, though. But that's where my point in all of this is if you have something that is 453 amino acids, and it's a bunch of amino acids that make up a polymer, what's the word that you typically use to describe this particular type of molecule? I'll give you a hint. It starts with a P. Add it to the chat if you know what I'm talking about. And it's not a peptide, but it's close to that. Give you another hint. It starts with a PR. A protein. Yeah, absolutely, a protein. So a protein, could everyone give me the letter O in the chat if you agree that a protein is a molecule, and an S in the chat if you don't agree that a protein is not a molecule? Yes, a protein is a molecule. Okay, that's an important thing to understand and kind of acknowledge and appreciate. Now, most enzymes are polymers of amino acids, also known as proteins. Okay, but so think about it as a protein that catalyzes a reaction is an example of an enzyme. Could you give me a one in the chat if you agree with that idea? Okay. Now, DNA molecules can be enzymes. These are known as DNA-zymes, also known as deoxyribozymes. These are synthetic. RNA molecules can be enzymes as well. RNA-zymes, also known as ribozymes. These are naturally occurring. And this is, if you've ever taken like an evolutionary biology class, this is maybe one of the things that you talked about in terms of the um, RNA world. Um, Basically, from an evolutionary standpoint, the first compounds that existed were RNAs, and they catalyzed reactions that gave rise to larger molecules, which gave rise to larger molecules, which gave rise to life. Now, what we are primarily going to be focusing on is enzymes that are polymers of amino acids. So there's a time where I will use the terms proteins interchangeably with enzymes. I want to stress, however, that not all proteins are enzymes and not all enzymes are proteins because some enzymes are made up of DNA and some enzymes are made up of RNA. Could you give me an 8 in the chat if you understand that? Okay, and any other number if you do not understand that. Okay, great. So I will do my best to try to distinguish between proteins and enzymes because hemoglobin is a protein, but it does not catalyze any sort of reaction. So is hemoglobin an enzyme? Y or N to the chat? Correct. It is not. Hemoglobin is not an enzyme. Is hemoglobin a protein? Y or N to the chat? Absolutely. Hemoglobin is a protein because it's a polymer of amino acids, but it is not an enzyme because an enzyme catalyzes a reaction. Now, when we talk about enzyme catalyzed reactions, it's important to know that, well, for our purposes, more times than not, we are going to be talking about polymers of amino acids that are enzymes. Now, the highlighted region here, could anyone add to the chat? What's this thing that I've got circled there? An alpha helix. Well, this is what I've got circled is helical, so that's an alpha helix. And then this right here would be kind of a beta or a random coil. I saw a couple people say beta sheet. Well, here is an example of a beta sheet. Does everyone see? It's kind of tough to see. And actually, see this right here? That is a region of a beta sheet. Here's another arrow. Anytime you see these arrows, I want you to think, okay, that's a beta sheet or a part of a beta sheet. Could you give me a 6 in the chat if, okay, I gotcha. All right, great. Now, when we talk about enzyme catalyzed reactions, um, I want you to understand that this right here is made up of amino acids. So what are the chances that there's, say, like an arginine there? Give me an R in the chat if it's possible. There's an arginine within that, or a glycine, or an alanine, or any of our proteinogenic amino acids. Absolutely. Same thing goes here. Same thing goes here. Same thing goes here. It's always important that you look at that and remember, okay, well, amino acids build these things up. They make these things. So what I'm looking at there is just a polymer of amino acids. Now, with that in mind, we need to know some of the players. The amino acid polymer is the enzyme represented by the ribbon structure, but what about the other thing? The other thing being in the middle. These would be what we know as our substrates. So the term ligand and substrate, they are not interchangeable, but in some respects they're comparable to one another. A ligand is what a protein will bind to and not catalyze a reaction, will not do anything to break or make any sort of bonds. A substrate, however, or substrates, these are molecules or compounds that an enzyme will bind onto and will do something to them. So when you see a substrate, what you should be kind of anticipating is something's going to change. Something will change here. Either these two molecules are going to be combined, or maybe one of them will be broken up. Something about that. So whenever you look at a model like this, it's always important to see, okay, what's my enzyme and what's my substrate? Could you say that again about the ligand's difference? Yeah, absolutely. A ligand will be bound by a protein. I'll say bound and released by a protein and no bonds, none of its bonds will change. I'll say, will break or be made. A ligand will be unchanged in its binding to a protein, whereas a substrate, something's going to happen with the substrate. Could everyone give me a 6 in the chat if you follow that and that difference between those two? Wonderful. Wonderful. Okay. So when we talk about enzymes, we are focused on and what we want to be kind of thoughtful of and aware of is the protein, or sorry, the enzyme and the substrate. Now, the context between these two, like what is it that's making this substrate interact with this enzyme? How is this enzyme making or breaking any sort of bonds? That's what we study whenever we are investigating, well, enzyme-driven reactions or enzyme catalyzed reactions. Now, what's the dynamic? What's happening there? The active site is the geographic spot or location on or within the enzyme that does the work. This is where the electrons are moved around and bonds are made and broken on that substrate. So the active site is kind of where the party happens in some respects. Now, when we look at the active site, what's important to know and recognize about the active site is, well, what are the amino acids that are there? So there's this term and this phrasing, active site residues. All amino acids within the active site have chemical properties that make them different. Okay, we already know that. When we talked about our proteinogenic amino acids, we talked about and classified them as polar and nonpolar. Okay, nonpolar residues like glycine or alanine or tryptophan. Okay, we know what those look like. Maybe there's lots of hydrocarbons there, leucine, isoleucine. And then you've got your polar residues like aspartic acid, glutamic acid, arginine, lysine, histidine. We've got all these different chemical properties of those different groups. We've talked about them in terms of the interactions that they can have. They can have hydrogen bonds or disulfide bonds or ionic interactions. These are all important things. Now, the active site residues are what are going, sorry, are the residues within the protein that interact with and make change happen on our substrate. Could you give me a 7 in the chat if you understand that? Okay. So our active site residues are the ones that make changes to our substrate. Arguably, the active site residues are the kind of cool and important ones. Okay. Now, let's get another poll question. If active site amino acids are responsible, think about your classification of amino acids. If your active site residues are responsible for pushing electrons, which of the following amino acids is most likely to be an active site residue? Okay, I haven't launched the question yet, so nobody's missing anything. Now I've launched it. Active site residues that can push electrons. What do we need to have for an amino acid to push electrons around? Well, if we think about our options here, creatine. Well, creatine is creatine an amino acid? Yes, it is. Is creatine a proteinogenic amino acid? No, it's not. So creatine isn't what we're looking for. Glycine and alanine. Could anyone add to the chat how those are classified? Are those polar residues or non-polar residues? Or yeah, these are non-polar residues. These are non-polar amino acids. Histidine is a polar positive residue. Okay. So we've got some sort of charge. We've got something that could potentially act as a really nice electrophile. Or it could act as a nucleophile. Histidine in particular could, which is kind of interesting and worth remembering. My point in all of this is that when we look at our different amino acids, these non-polar residues, that's like saying, hey, how good is a methyl group at pushing electrons around? Spoiler, not very good. Because what do we know about that methyl group? CH, H, H. I always think of a methyl group as similar to like the most boring person that you know. It's like, oh, cool, you got 4 bonds. So what do we talk about? Great. Well, thank you. Goodbye. This isn't exciting. Meanwhile, histidine, on the other hand, we got histidine looks like a house. Histidine is going to have a positive charge here. It's like I'm a ring, I'm a five-membered ring, which what do we know about rings? Six-membered rings are stabilized, but I'm a five-membered ring. Spectacular. You know what I'm going to do? I'm going to go out and find some electrons. It's going to be great. Okay, so my point is the correct answer is D. And somebody answered B at the last moment. I am going to presume I'm going to close the poll now. Okay. So now this is a visual of substrate binding and an active site. Typically, whenever we look at these sorts of models or a protein structure, it's pretty common to depict the substrate and the enzyme with different kind of representations. So this substrate is going to be your typical stick model or your simple skeletal structure, whereas your enzyme, this particular enzyme is an enzyme known as lysozyme. This is going to be represented with a space-filled model. And I'm sharing this as just a, hey, when you see this, here's what you should anticipate. Now, I'm going to go back here and show you that this right here, well, this is shown with what's known as a ribbon diagram. So I'm going to go ahead and label that ribbon diagram or ribbon model. And our substrate here is represented as a ball and stick representation. So there we go, space filled. The different residues that are important are highlighted also in this model with different colors. We've got our substrate binding site, and then we've got highlighted in red the particular active site residues that are important for making or breaking any sorts of bonds. So this is something that you kind of have to be thoughtful and cautious of. This whole thing is going to be in contact with the substrate, but not all of these residues are involved in making and breaking bonds. We'll get into more of those a little bit later on and go into some more specifics about that. But for the time being, I want you to look at this and say, oh, okay, well, there's maybe two or three active site residues, but then there's a lot of other things that are involved in like making that action happen because they will help the substrate get into the proper position. Oh, sorry. Okay. Is the active site where both ligands and substrates combined or only substrates? Okay, I'm happy that you asked this question. If you're talking about an enzyme, don't talk about ligands because when you're talking about an enzyme, the binding partner of an enzyme is the substrate. So only a substrate is going to bind in the active site. When we talk about a protein ligand interaction, then we will talk about a ligand binding site or a binding site and the protein. So like maybe the blue holds in and the rest does the work. The red does the work. Absolutely. That's absolutely what happens. In this particular enzyme, lysozyme, we'll get into that a little bit more later on and how it treats the substrate it's almost like a conveyor belt where it loads up the substrate, my left arm being the substrate here, and the active site cuts, and then it shifts it down, cuts, shifts it down, cuts. So we'll get into that later on, but for the time being, yes, that's a great understanding. Okay, so what I want you guys to do is whenever we're going through this sort of stuff, be thinking enzyme substrate, enzyme substrate. And actually go ahead and start using E and S versus P and L. P and L would be protein ligand, E and S would be, well, our enzyme and our substrate. Now, with that in mind, what do you think you would note the complex of an enzyme and substrate when they come together? How would you simply write that out? If E is enzyme and S is substrate, what would you refer to that complex as? Maybe ES? Does that sound good? Give me a 5 in the chat if that sounds good. ES. Wonderful. Wonderful. Just as an aside here, I'm drinking a Diet Dr. Pepper and I I added protein powder to it, some whey protein, and it's pretty much as tasty as you would think. But anyway, that's lunch today. Okay, so moving forward, enzyme substrate binding. Here, what we have is our ball and stick representation of substrate and substrate. Spoiler, it is possible for an enzyme to have two different substrates, and that's fine. Now, here's our enzyme, our substrate A, our substrate B. This is just kind of giving you the logistics of what's taking place. Our enzyme will bind our two substrates. If it binds two substrates, this will form what's known as the ES complex. I want you to know that as ES, as one complete thing. So that means the enzyme has bound to the two substrates, or in some cases, individual substrates. The enzyme will bring these two things in close enough proximity where a reaction is going to take place between our two different substrates. This is denoted as ES star. This is where our reaction's taken place, but our substrates are still bound by our enzyme. Substrate A has become C, B has become D. And to kind of show what's taking place, I want to take you back for a second. Here's our substrate A. Could anyone tell me what color this is right here where I put my X? Maybe like pink. Good. That pink molecule is bound to substrate A. That's a part of substrate A. And this enzyme, what it's going to do is it's going to move that part of substrate A onto substrate B. And we see that. Now we've got substrate B that is different because it's taken on a part of C or of part A. So C, answer Y or N. Is C the exact same molecule as substrate A? Y or N? Is it the exact same? No, it's not, because it lost that pink kind of part of it. Now, product D, is that the exact same as substrate B? No, it is not. So our two products, C and D, have been released. Our free enzyme is now ready to do it again. Okay, and this is depicted at E plus P. Our free enzyme represented as E and our free product represented as P. Could everyone give me a 9 in the chat if they follow that kind of sequence? Free enzyme, okay, ES, ES star, E plus P. Wonderful. Now, this is a rundown of all of that. E plus S, free enzyme, free substrate. ES star is where the reaction is taking place. EP is where the products are present. And then E plus P is where the products have been released. Now, when it comes to an enzyme, we're very interested in the mechanics of it all. We're interested in the binding specificity. So does an enzyme only bind to one specific substrate, or could it bind a whole host of different or similar substrates? We also want to know about how flexible this enzyme is, and we want to know about the electronic environment. So are there a bunch of positively charged groups and negatively charged groups and ionic bonds that stabilize it? Or what ultimately is it that stabilizes that structure and that enzyme? And are there any coenzymes? So are there small molecules that assist in that reaction? So small non-substrate molecules that assist in the reaction taking place. Now, here are some models. So this would be our enzyme, and here's a substrate. So a simple catalytic model is that our substrate enters the active site of the enzyme, and the enzyme basically closes off and clamps down around the substrate, forming what's known as an induced fit. So whenever this substrate binds, well, this fit goes on to take place. Now, there is something known as the Fisher mechanism in terms of specificity. The Fisher mechanism is an idea that the substrate, the shape of the substrate, and the composition of the substrate fits perfectly in to the pocket of the enzyme. And that's how we get this ES complex. Okay. Um, Seems like it makes sense. It's almost like a lock and key. A key fits perfectly into that lock. There's grooves on that key, and then there's indentions in that key, and there's a complementary side of that on your enzyme. Now, that could you give me a six in the chat if that makes sense? Okay. Now, this is the Fisher mechanism. This is kind of rigid, and this is just kind of a theory as to how it works. There's also a mechanism that was deduced by a guy named, I believe it was Roger Koshland. And he proposed that, well, I think that what happens is we've got an enzyme that's made up of a bunch of different amino acids, and the shape of it is somewhat fluid. And what happens is the active site will make minor modifications and make minor changes in order to adapt to the shape of that substrate. So here, our enzyme E binds our substrate. And if you look at the shape of that substrate, well, it roughly matches the footprint found within the enzyme. But then when the ES complex forms, it's almost like that enzyme kind of more tightly wrapped on that substrate. Could everyone give me a seven in the chat if that makes sense? Now, these two models are not necessarily, they are a little bit at odds with one another, but this is the sort of thing that you can't say, well, one is true and the other is false. One is good and one is bad. Are there some enzymes that support this model? Yeah. Are there enzymes that support this model? Yes. I want you guys to know that the active site is something that in some enzymes will adjust itself to fit the substrate, and then the active site will also, well, not necessarily adjust itself to fit the substrate in other enzymes. Okay, now this is kind of your enzyme substrate interaction. Some of them it's rigid and some of it's a little bit more mobile. Now, in terms of what an enzyme is doing and how it's doing it, what we're interested in is free energy. And has anyone seen something like this before? This sort of free energy diagram? Give me a one in the chat if you've seen something like this before. Okay. You probably saw it when you were talking in Gen Chem 2, where you looked at a reaction and, well, we see the reaction progress. This side represents your substrates, and this side represents your products. Okay. Now, in order for a reaction to take place, your substrates are typically going to be at higher energy than your products are. Okay, high energy to low energy. Now, how do we get from here to here? Well, you have to go through what's known as a transition state. And the free energy of that transition state, it's almost like, well, your substrates or your reactants, well, they have to kind of be positioned and contorted to undergo these sorts of changes. Now, here is the delta G of our uncatalyzed transition state. As soon as you get to that delta G, as soon as the free energy changes have taken place, well, this is the highest free energy of this reaction. What's going to happen is your reactants that reach this free energy state, they're ultimately going to go down here to reach a lower free energy because at that lower free energy, it's a little bit more stable. This is also known as your activation energy to catalyze this reaction. Now, what I need you to take away from this is the delta G of your initial state, your transition state, and your final state. And then the delta G, the total free energy of the reaction. Okay, the total free energy of your reaction is the free energy difference between your reactants or your substrates and your products. Now, When it comes to an enzyme catalyzed reaction, well, what's going to happen is an enzyme is going to lower the transition state or activation energy. The total free energy of the reaction, the delta G total, is not going to be changed by an enzyme. What's going to be changed is the activation state or the activation energy. So an enzyme, the way I always kind of characterized it, and I think that one of my undergrad professors said this, was basically an enzyme is just going to make the hill that needs to be climbed a little bit lower. So an enzyme lowers activation energy. So that's what an enzyme is going to do. That's what we're kind of banking on. The enzyme will position the substrates in a perfect way so that the groups that are going to react are in close proximity to one another. An enzyme will cause a molecule or a substrate to bind in a little bit more favorable position so that the reaction's more likely to take place. So energy's lower activation energy. They catalyze reversible reactions because, the as soon as you reach that transition state or that activation energy point, the reaction could go backwards. It could go forward. More than likely, it's going to go forward, but an enzyme can catalyze a reverse reaction and a forward reaction. So an enzyme will catalyze reversible reactions. They do not change the overall free energy or the overall energy, and they do not change equilibrium concentrations. They will speed up achieving equilibrium. So we're going to get to that point a little bit quickly or more quickly with an enzyme. Now, enzyme reactions. Okay, so what I'm going to do is actually go past this one. And what I want to talk, okay, no. Okay. Does anyone have any questions about this right here? Give me a 5 in the chat if you feel all right about it. And any other number if you have a question. Okay, great. So, here we go. An enzymatic reaction, we have reactant A or substrate A going to product B. Now, at equilibrium, the concentration of A at a given time is equal to the concentration of A 5 seconds, 5 minutes from there, because we're at equilibrium. Those concentrations of reactant A or substrate A, that's not going to change because we are at equilibrium. Similarly, the product, well, at equilibrium and 5 minutes later, the concentration of B is going to be unchanged. At any amount of time X after equilibrium has been reached, the concentration of A is going to be the same. The concentration of B is going to be the exact same. Does everyone give me a 7 in the chat if you follow that? Any other number if you do not follow that? This is the sort of thing that where people run into a problem, in my experience, is that they say, oh yeah, but what about relating A and B to one another? What can we say about the concentration of A and B when we're at equilibrium? Well, we can't say a whole lot beyond, well, excuse me, we have an equilibrium constant, K, or a KEQ. What we can't say is that the concentration of A is equal to the concentration of B. Unless delta G is at 0, it's wrong to say A is equal to B. Now, when it comes to substrate binding, this is important to know because there's a number of different ways in which enzymes can catalyze a reaction. An enzyme can have a single substrate, and that single substrate can be converted to a single product. Here we've got A goes to B. An enzyme could also have multiple substrates and or it could have a single substrate and multiple products. It could have multiple substrates and a single product, or it could have multiple substrates and multiple products. Now, I bring and highlight this one because this is where we're going to spend a little bit of time. So A and B go to C and D. That's the model that we already kind of saw. Now, the different outcomes from this are going to be, well, it could be ordered, random, or ping pong. Huh? Ordered, random, or ping pong all relate to the binding and release of our substrates and our products. The first one that I want to talk about is the ordered multiple substrate binding. So this ordered mechanism, I'll call this as an ordered mechanism. I don't know about you, but whenever I think of ordered, I think of organization. I think of a sequence, like get into line alphabetical order. The sequence matters. So in an ordered mechanism, well, we've got an enzyme, specifically lactate dehydrogenase. And lactate dehydrogenase has one, two substrates, and one, two products. Now, what's significant about lactate dehydrogenase is that NADH, this is a molecule known as nicotinamide adenine dinucleotide, that has to bind first. So essentially, if pyruvate, the other substrate, and I'll actually just simplify these, I'll say A and B, C and D. If B binds to this first, the reaction's basically not going to take place. A has to bind first. Now, can you give me a 9 in the chat if that makes sense? Okay. Now, a random reaction. Creatine kinase. Creatine kinase is an enzyme that's very important if you consume lots of creatine because it kind of supplements your ATP pools. Creatine kinase, well, it has two substrates, creatine and ATP, and it produces creatine phosphate and ADP. Okay? The sequence that they order or that they bind does not matter. Creatine can bind creatine kinase first, and then ATP will bind. So first and second. Or ATP will bind, and then creatine will bind. Doesn't really matter. In either case, creatine phosphate and ADP are going to be produced. The sequence of those that they are produced also does not matter. Okay, so no order is actually required for that. So we've got order or random. The question that's basically being asked, does sequence matter? That's our question. And if the answer is yes, it's an ordered reaction. It's no, then it's random. Okay. Now, the next mechanism or type of mechanism is known as ping pong catalysis. And here what we have is an enzyme that will bind substrate A and substrate B. And it will produce product B and product E. Okay, now basically what's happening here, there's another way that I like to depict. Okay, so, sorry, let me erase that. So our substrate A has this carbonyl oxygen. Our enzyme has this amine group. This enzyme will take this amine group and place it onto our substrate and in turn take that carbonyl oxygen. So our product is going to be basically our aminated substrate A. Could everyone give me a 1 in the chat if they follow that? So I always kind of think about ping pong catalysis as having two separate reactions. Reaction 1, reaction 2. So for reaction 1, do they bind at the same time? Melanie, I'm sorry about that. E is our enzyme. So our first substrate is A. So A is a substrate and um, C is a substrate. So substrates, let me write this out. Here are A and C. Products are B and D. Could everyone give me a 9 in the chat if that makes sense, that A and C are the substrates and B and D are the products? Okay. So reaction number one, our enzyme binds substrate A and transfers an amine group onto substrate A and in turn takes that carbonyl oxygen. So our first product is released. So reaction number one releases product one. Okay, now the next part about this is I'm going to erase all this. And what I want to ask you is this the exact same as this? Y or N to the chat? No, it's not. So our enzyme is aminated, starts out like this. And then it ends like this at the end of reaction number one. So then if we go down to reaction number two, our enzyme has this carbonyl oxygen. And what it is going to do is going to bind substrate C with an aminated C, our second substrate, and then it's going to change, or it's going to take that carbonyl oxygen and transfer it onto substrate C, and we're going to get product D, or our second product. And we're also going to get our aminated enzyme. Okay, so there's a couple of statements that I want you to know about this. So reaction number two releases product two. Oh. Okay. Let me ask you this. Can ENH2. ENH2 is the form of our enzyme that binds substrate A. Okay? Give me a 1 in the chat if you understand that. That ENH2 is the form of our enzyme that binds substrate A. Okay? Now, a little bit ago, everyone seemingly agreed that ENH2 and E carbonyl oxygen, or EO, this and this are different. And I agree with that. They are different. Now, my question for you is, can ENH2 bind substrate C? Give me a 0 in the chat if it cannot, and an 8 in the chat if it can. Correct, it cannot. So ENH2 and E, they are not the same. So they are different forms of the same enzyme. Say this, different forms of enzyme. So in a ping pong mechanism, your enzyme E changes. And basically has different forms. Can both B and C react with B and C? Okay, so EO. That is a great question, Melanie. Yes, it can. Now that adds like a wrinkle of complexity to it. It can because of the fact that, well, it could catalyze the reversible reaction. Now, what I want you guys to know about this, and what I want you to walk away from this slide with, is I want you to know that our enzyme changes and has a different form in order to, or it has an intermediate form that it has to get to in order to make our two products. Could you give me a five in the chat if you follow that? Okay. So the biggest part about this is that in a ping pong mechanism, a different form of the enzyme will emerge. Now, is this why some enzymes work differently when in different tissues? That is okay. So, is this why some enzymes work differently when in different tissues? That's a part of it. No, I don't know. Um, if an enzyme's going to work in a so for example, hexokinase is responsible for breaking down sugars like glucose and mannose. Glucokinase is found uniquely in the liver. Hexokinase is found in basically every tissue. The difference between them has to do with their affinities for different sugars and therefore availability of different sugars. So if you took an enzyme and put it into kind of a foreign place, it's probably going to act different because of the environmental conditions, maybe more than what we just kind of went over. But that's the best answer I can come up with. Would there be competition between B and C, or we do not have to worry about that? That would There would be, but that's ultimately going to be based on the concentrations. But for our purposes, we don't really need to stress about that because we can kind of look at this. Okay, well, it's going in this direction. E, our enzyme, is going to change a little bit. So the enzyme switches what it was bonded to. Yeah. E NH2 versus E carbonyl oxygen. Yeah. Okay. So this is just an example of that playing out. So our enzyme is going to ultimately, our transaminase is our enzyme, and ultimately what it's going to do is it's going to take a molecule and another molecule, our two substrates, and it's going to produce two different products from all of this. It's going to remove, or it's going to move an amine group to one molecule and carbonyl to another. Now, how do we evaluate enzymes? This last bit, which we're going to be able to, oh, I thought it was 137. Dang it. When we study enzymes, we look at Michaelis-Menten kinetics. So this slide right here, I'm not going to go beyond 150. Don't worry about that, anyone. If you got to go, though, you got to go. It's being recorded. This graph shows us concentration. It doesn't designate one specific thing. Instead, the lines represent different things. So our free substrate. This slide is basically, hey, we're transitioning. There's different models in which enzymes bind substrates and release products. Now we're going to talk about how do we analyze an enzyme? What's important about an enzyme and its analysis? When we look at the substrate concentration and the product concentration in a given chemical reaction or in any reaction, substrate or reactant concentration is going to go down and product concentration is going to go up. Give me a five in the chat if you agree with that idea. Reactant concentration goes down, product goes up. Great. And ultimately, it will reach an equilibrium. An enzyme, a free enzyme. Well, when we talk about enzymes, we talk about enzymes or as a part of the enzyme substrate complex. Our free enzyme concentration will start at time point 0. It's at its highest. It will drop down. Free enzyme will drop down. Yes, because that free enzyme is binding the free substrate, which is why our ES complex concentration will go up, as depicted by this kind of grayish line. Over time, as this reaction takes place, so this is kind of time point 0 on into infinity, well, our enzyme substrate concentration will go up and then it will go down. Our enzyme concentration, our free enzyme concentration will start high, go low, go back up. The free enzyme concentration and the free product concentration will be going up in pairs or going up kind of together. Now, these are all the different players of this. When it comes to studying an enzyme, what we are interested in, and this is my last slide, is we are interested in what is known as the steady state, where our enzyme concentration and enzyme substrate concentrations remain relatively constant. That is the window in which we're going to work. Now, that is where I'm going to stop. I'm going to say thank you guys very much for coming to class. I'm going to stop my recording